Good morning, everyone. Welcome. My name is Laura Riley. I'm the coordinator of Chicago Wilderness, and we're happy to welcome you all today. We'll just give it a few more minutes while folks join us and we let them in. Please keep yourself on mute for the time being. You can drop any questions into the chat. Today's event will be recorded and will be posted on the website, as well as it will be shared out with everyone who registered for today. So you will receive a recording and a PDF of the presentation from today.
Okay, I see there are still people joining, so maybe we give it another minute. My name is Thorsten Lumsch. I'm Vice President at the Field Museum, and it's my pleasure to welcome you all to this joint presentation of the Chicago Wilderness Café and the Watson Armour seminar series of the Field Museum on the 2022 Biota Awards. Maybe we can have the next slide, please. The Field Museum acknowledges that it resides within the traditional homeland of the three Fires Confederacy, the Ojibwe, Odawa, and Potawatomi. The museum recognizes and is grateful for the original peoples who laid the foundation for the city of Chicago and for the diverse indigenous nations that reside in Chicago today. Let me start by thanking the Armour Lecture Series Committee and the Chicago Wilderness for putting this conference on today. And special thanks to the Walder Foundation for this new award supporting biodiversity science and conservation in the Chicagoland region. The Field Museum's mission is to connect all of us to the natural world and the human story. This work is particularly important here in the Chicago regions, where we are deepening and expanding our biodiversity science and conservation. I'm very pleased to introduce these remarkable young biodiversity scientists who are some of this year's Biota awardees. And I'm looking forward to hearing from them and to hearing from you about how these projects relate to your work and your interests. Without further ado, I would like to ask Jack Westwood from the Walder Foundation to say a few words. Thank you.
Thank you, Thorsten. And thank you to Chicago Wilderness and the Field Museum for hosting this webinar today. It's really great to be here, and I too am really looking forward to hearing from three of this year's awardees. My name is Jack Westwood, and I'm the Program Director for Environmental Sustainability at the Walder Foundation. Walder Foundation is a family foundation that's based in Skokie. It's been around for about three, 4 years now, and a very place-based foundation. Across all of our programs, we're really focused on the Chicago region, and environmental sustainability is one of those programs. We started the Biota Awards last year to support biodiversity research in the Chicago region in a couple of ways. One was to provide research funding to early career scientists at a really critical stage in their career, where we felt like a boost to their research programs would really help them on their career trajectories. And the second was to really encourage and support more links and connections between the great research happening and kind of on the ground conservation community, and trying to help research deliver on the ground results. So in the first year of the awards program, we made 5 awards in total. These awards are a total of $300,000 each, spread across 3 years in research funding. And we were really excited about the range of projects and proposals that we received, and in particular the awards we made. Not featured today in this webinar are 2 other awardees, Becky Barak from Chicago Botanic Garden who's working on a project looking at lawn alternatives and how they can support biodiversity, and that's in partnership with the Chicago Park District. And also Megan Midgley from the Morton Arboretum, who's working on burn pile scars in forest preserves and trying to understand the impacts of burn piles from brush pile burning on soil biodiversity and on restoration ecology. I wanted to highlight that the program is currently open for applications for this year. The deadline is October 3rd, and you can go to biotaAwards.org for more information about that. And there's some information on that site about how to get in touch with us as well if you have any questions. I will say, just highlight one key change from last year is that this year the awards are open to the whole state of Illinois. So any researchers that are based in Illinois are eligible to apply, but there must be some kind of connection to Chicago or the Chicago region through those projects. And again, feel free to be in touch with any questions about that. That's all I have to say to you today. Really excited to hear the presentations, and I'd like to introduce the first awardee, which is Adam Ferguson from the Field Museum. Thanks.
Great. Thanks, Jack. Thanks, everyone. Thanks for organizing this and inviting us to talk a little bit about the Biota project that was funded for this year. So I'll start my timer and share my screen. All right, hopefully my presentation is showing up. All right, great, thanks. So the title of our project was Small Mammal and Pathogen Diversity in Southside Parks in Chicago, Implications for Human Health and Well-Being. And one of the things we really wanted to emphasize with this project was to try to implement a One Health approach to understanding zoonotic pathogens and green spaces in the south side of Chicago. So I just want to start by thanking this project wouldn't have been possible without the Walder Foundation and the Biota Award in particular. But this is, even though I'm talking today, a huge collaborative effort across multiple institutions here in the Chicagoland region. And I just want to acknowledge our major partners and collaborators, including the Forest Preserves of Cook County, Chicago Park District, Chicago State University, Wheaton College, UIUC, especially their medical entomology program, and the Lincoln Park Zoo. So as I said, this is a very large collaborative project, and each partner is bringing unique aspects to the different objectives of the project. I'm not going to go through each one of these, but these are just highlighting some of the ways that the different partners are participating and bringing in expertise to make this a holistic approach to understanding pathogens and small mammals in the system. So first steps of the project was to get the whole team together, right? So we'd been in communication before, but once we got funded, we were together. We're using the Ford Calumet Environmental Center, which is based at Big Marsh Park, run by the Chicago Parks District as our headquarters. And so it's an amazing meeting space and an amazing park. So we had our first meeting there. And of course, the priority was coming up with a catchy acronym, which I learned long ago, good projects always have. So we spent some fun times brainstorming on acronyms and came up with the CAPTURE project, which stands for the Calumet Animal Pathogen Urban Research Experience. So it's CAPTURE in the sense that we are putting hands-on and trapping small mammals to look at pathogens, but we really wanted to emphasize this experience part, that this should be an experience. And this is a very student-centric, student-focused project, right? So we have Zoe Leone, who just joined us as the field sites coordinator for this year. And then Angie Bahina is going to be joining us as the field sites coordinator, running the grids and sampling for years 2 and 3, but also pursuing her master's in relation to the project at Chicago State University. We have funding for interns through Chicago State University, and then we hope to involve as many volunteers and students as possible. So the amazing thing about the flexibility of these funds is we're able to support these students fully to pursue their own research while they also help contribute to the bigger picture and be part of this project. So briefly, the research questions, there are 4 kind of major research questions we were interested in answering with this project. The first is, how does small mammal diversity vary across natural areas in Chicago's South Side? Focusing mostly on ticks and tick-borne pathogens, looking at how tick populations and infestation rates on these small mammals might vary across different land use types and the different species of rodents and or shrews that we find in this area. And then third, how do pathogens associated with both these small mammals and their vector hosts, the vectors vary in response to those different land use practices? And then we also really wanted to get a kind of a gauge on community members' perceptions of rodents, zoonoses, and other factors related to green spaces in their own backyard, with the idea being that green spaces are important, but sometimes understanding the community context and the value they bring is really important, especially in regards to things such as maybe of concern like Lyme disease or other pathogens or zoonoses. So rather than reinvent the wheel, we're also kind of trying to adopt NEON protocols, which is the National Ecological Observatory Network sampling protocols, where they use small mammals and vectors as ticks as sentinels of environmental change through time. And if you can see from the map here, Chicago is kind of between two of the major regions and kind of a unique system. They don't have any urban sites with the NEON protocol, so we've been working with them to kind of adapt and develop methods from their long-term sampling that allows for repeatability to examine these patterns in ticks and small mammal diversity. So we're focusing on 4 major sites. The sites have changed slightly, but 4 major sites in and around the Chicago State University for ease of access. That's Big Marsh. We're also working at Eggers Grove, Beaubien Woods, Dan Ryan Woods, and then we've augmented with some other CPD sites, including Hegwish and some of these other parks that are in this region. The sampling strategies focusing on, again, mentioned small mammals, tick vectors, and then molecular pathobiological screening in the lab. I'm not going to go into great detail, but the beauty of having this 3 years is we're working out the kinks in year 1 with our pilot work now, but the idea is to run 4 trapping grids per site. We've reduced the trapping grids to 81 to fit into these smaller areas in an urban context. And we're stratifying these sampling grids by habitat type, with 8 woodland, a balanced design of 8 woodland and 8 open and or prairie habitats. We're using a combination of capture, mark, recapture, non-lethal and lethal techniques to estimate small mammal diversity, both abundance and richness. And then we mentioned the field team, and we also have a lab team following a cohort model that'll help with the screening of the pathogens. There's 4 kind of major pathogens, or 3 major pathogens of interest we're focusing on, but we're also doing de novo assessments of other potential unknown pathogens. That's raccoon roundworm, Rocky Mountain spotted fever is the disease caused by rickettsia, and then Lyme disease and other Borrelia bacteria species. So in terms of the project outcomes, we hope to, beyond the research and scientific value of the questions we're asking, collect actionable data for land managers, community members, and public health officials to somewhat establish a longitudinal, long-term sampling regime that results in specimens and material being permanently archived at FMH for future use and looking at changes in time. Student-focused training on One Health, wildlife biology, and museum science. And then most importantly, and with the Chicago Wilderness here as an example, enhanced collaborations aimed at addressing these local issues of conservation and community engagement and local green spaces. And that's a picture about 30 minutes ago. We just finished the first specimen of the project. There is a short-tailed shrew that we salvaged. We found dead when we wwe were setting our grids at Big Marsh. So data's coming in. It's not rolling in, but it's in. And with that, I just want to thank, we have lots of partners, lots of support, and a really collaborative group. So I'm just if I forgot anyone, my apologies, but put as many of the folks up there at the different organizations that are working as part of this project. And thank you for listening. All right. And with that, I will turn it over to the next awardees, Sara, Ruane, and Michelle Thompson.
Hi, everybody. So let's just share our screen, share sound, because we have a little bit of sound here. And hopefully we're all seeing this. Share my whole, sorry, it's asking me for a password. Please let that work. All right, my speaker is not working. Uh-oh. Can everybody hear me right now? Thumbs up.
You sound great.
All right. Well, we have sound. It may or may not work. So we'll see when we get to that slide, but we don't want to hold things up.
All right. Hey, everyone. Thanks for joining us today. So I'm Michelle and this is Sara. And today we're going to talk a little bit about our research and outreach work on resurveys of herpetofauna, which are amphibians and reptiles in Will County. We'll talk a little bit about project goals, what we've been up to since we've been funded in February, and what the future holds for this project.
So one thing we want to talk a little bit about is resurveys in general and why bother doing them, whether that's in Will County, Illinois, or anywhere in the country. And one of the main reasons is determining change over time. So this can be due to things like anthropogenic change, so human-induced change, or it could be natural changes. It could be due to introductions of invasive species. There's a lot of things that can change what's been in a place historically with what's there currently. And one of the things that's really important is that it allows you to inform management systems. So that could be, for example, in our case, the Will County Forest Preserves Management Team. And Will County was last surveyed in the 1990s and 2000s, early 2000s, specifically for reptiles and amphibians, but it wasn't done in a particularly systematic way. It was more just casual encounters. And a couple of reasons we want to look at Will County in particular, which is highlighted there on our screen, both using a Google map and then just showing all the main counties of the Chicagoland area, is because it actually has the greatest herpetological diversity. So the most numbers of reptile and amphibian species in all the counties surrounding Chicago. And combined with that, it's one of the fastest growing counties in not just Illinois, but actually in the United States in terms of populations. One of the things that sort of inspired us to focus on this was the Grinnell Resurvey, which is a big NSF-funded resurvey out of California, using all these historical field notes from Grinnell and going back to those sites and seeing how things have changed over time. And so when we were putting together this grant, that was one of the things that was our inspiration, because that has been a really, really fabulous team of field work that's shown a lot about how things have changed for organismal diversity over time.
All right, and so this is collaborative work. And so our main fieldwork partners here are the Will County Forest Preserve District. And this was, although Sara and I had the aim of doing this resurvey work, we really co-developed the details and the priorities of this work with the Forest Preserve District. And this is an important point because we wanted to make sure that the results were applicable to conservation and land management. And so most of the field work is a collaboration between Sara and I, also many interns that at least 5 that run the field work from year to year. And we also have involved collaborators and contractors, Tom Anton and Dave Mauger, and these are the biologists that originally did the resurvey work. So we get all this great information from them. And they also get to interact with the interns and teach them many different things. And as well as our main contact, Becky Blankenship at the Forest Preserve District.
Yeah, Becky has really helped us figure out what preserves we wanted to focus on when we first started this work. And so some of the goals of this project, so we really got things moving along in May 2022, which is when some of these photos are from, but we wanted to look at the role of the preserves in conservation for urban or semi-urban biodiversity. Are these reserves and preserves really holding a lot of biodiversity that you're not getting otherwise in adjacent areas? Collaborate, as Michelle just said, with the Forest Preserves Management Team in order to get the most out of it for everyone. Some of the things we're looking at are species abundance and distributions compared to these older surveys. We're creating a genetic repository, taking tissue samples from individuals of every species encountered from every site we go to. And then there's a lot of training and outreach as part of this project as well with the interns that we talked about.
All right, so since we were funded earlier this year, our current status is that we went through the prioritization process with the Will County Forest Preserve District to prioritize the 82. There's many different forest preserve sites, so we kind of have them in different priorities of how much effort we're putting into each site and where we're focusing. We hired 5 college-age interns, which we broke up then into teams that help us run the east side of the county and the west side of the county surveys. So interns also get, some of the lead interns get practice in like a leadership and mentorship role as well. And over the course of the summer, these interns mostly, as well as Sara and I, have collected over 230 non-lethal tissue samples. So these are small pieces of tissue that we take from each animal, and then we release the animals and they go on their way. And this helps us build a genetic library for future use and research. We collected over 45 voucher specimens, which are deposited here in the Field Museum. We also have audio recorders out. Some of these species are really hard to see, but they're really noisy. So like frogs that call. And so we have about 4+ months of recording data. And this helps us identify not only what species are at the sites, But because frogs call when they're trying to breed or mate, it gives us information on reproductive seasonality. And plus we have many different observations with photo vouchers. And so these offer other opportunities for volunteers or We Dig Bio projects to engage with other volunteers in the community to digitize these data.
So just to show and highlight some of the work that's been done over the summer, these are the 5 interns that we have. And I think Michelle and I are also in a couple of these photos, but this is really about the interns. They're out there 4 days a week, checking traps, looking under cover boards, doing visual surveys. And so they are primarily responsible for all of those data that Michelle just talked about. And they've been just fantastic. And not only are they out in the field, but we also have given them some opportunities to come into the lab and see the other side of the work where we're prepping specimens and getting them ready to deposit here in the collection. And another thing that the interns have done, not those 5 interns, but there's a digital media internship program here at the museum. And when we were approached about potentially working with them, one of the things that we wanted to do and wrote about in the Walder grant that we submitted originally was creating field guides for the area, not specifically even Will County, but just the Chicagoland area that we could print and put out. And that was a big part of our budget, actually. And so this opportunity with the digital intern program here at the museum, 3 high school students got together and we sort of guided them in what it was we wanted it to look like. And then the team working with them who run the program really helped them learn how to do the digitization of a bunch of photographs. And there's still more edits to be made and the content needs to be adjusted a bit, but ultimately this is a proof of, or a draft of what the guide's gonna look like with one of the species highlighted a little bigger so you can see it. We're gonna get this printed out and laminated. It's gonna be a flip guide and we'll be giving lots of these to different people or organizations that are involved. So if you want one, just let us know. They're gonna be freely available to anybody who wants them. And we'll also put them online with the other museum guides that have already been made, so people anywhere can download them or use them if they want to.
All right, so our bioacoustics work, or our frog call data that we've compiled so far is about 4 months long. And again, like I was saying, this helps us ID what's there, gives us a little more information on when their frogs are trying to produce and reproduce, and does that change from year to year. But also we are in collaboration with the Field Museum guides to build a bioacoustics guide with this data. So not only are we creating a guide where people have sort of a visual, what are they seeing, but we can create a guide so that people know what are they hearing. And so here's some examples of some of the calls. This is a gray tree frog. And what you're seeing here is a spectrogram. It's just this little visualization of the call that you're gonna hear.
Can people hear it? Thumbs up. All right, great.
So this other one is the green frog, and that one's sort of like a banjo. It's probably familiar to people. So the idea is to compile all this together, so when people are out and hearing calls, they know what they're hearing.
And so some of the things we have coming up now that we're wrapping up this first field season is finalizing those flip guides and getting those ready for distribution. We're going to be entering and collating all these data that we have and starting to analyze and look for some trends in our data sets. We weren't able to start targeted amphibian surveying due to just timing with the money and getting the project going and permits. But for spring of 2023 and 2024, we are going to be able to get out there in March and April when amphibians are breeding and get a lot more data there. And of course, we're going to continue these general surveys and collecting during 2023 and 2024, focusing on some of the same preserves, but also adding in new preserves. And we're going to start getting that genetic barcode library put together. And again, that's going to be something that will be useful to us in some of the publications we're planning, but it's also going to be available to scientists and researchers worldwide through general loan policies here at the museum.
So thank you very much. We enjoyed sharing our work with you today, and we'll pass it on to Noe.
Awesome. Let's see, we can get this going here. Okay, can everybody see? Can you get a verbal yes, please?
Yes.
Okay, is this the whole screen
No.
or is it both screens?
I
Actually,
only see you. I don't see your presentation yet, Noah.
Yeah.
Okay, good call. Let me see, let's try it again. Sorry about that. All right, how about now?
perfect, but you need to do presenter.
It's slow, give me 2 seconds here. Of course it froze up. Let me see, here we go. Good now?
I think we're seeing your view rather than the presentation or it's like you need to switch your screens.
Try it again,
And
try it again, one more time.
slideshow.
Here we go. There we go. Can you see me now?
I think it's good.
Excellent. Okay. Bueno, muy buenos días, damas y caballeros. Good morning, ladies and gentlemen. My name is Noé de la Sancha. I am a research associate at the Field Museum. And so I really am thankful that I had the privilege to be included in this awesome group of people. And so right now I've just accepted a new position at DePaul University, which I'm pretty excited about. And so this is part of the ongoing research that I've been doing to some degree at the Field Museum for over 10 years now, which is kind of interesting how quickly time goes. So I am the only person right now that is doing work outside of the Chicagoland area. And this is sort of ongoing research that we've been doing in Paraguay. And so I'm going to try to outline what we're doing here. So today I'm going to be talking about our research that deals with, let's see. Okay, with mammals and deforestation. So when we think about mammals, just like many vertebrates throughout the world, we find that the largest number of species of mammals on the planet is around the tropics. This is where we also see the most phylogenetic diversity. Now, as we all know, we're currently in the 6th mass extinction, which is primarily driven by human-driven causes. One of the main reasons why we're losing, especially mammal species in the world, is habitat loss and habitat fragmentation. This is also important because one of the things to keep in mind about the tropics is this is where we have the highest number of newly described species and where we know the least about mammals, generally speaking. This is where I do most of my research. This is the Atlantic forest of South America. If you can see, it goes from about northeastern Brazil, all along the coast of Brazil into Argentina, into Paraguay, which is where I do most of my research. Big picture objectives are to identify multiple dimensions of biodiversity as a function of deforestation. We want to understand how these dimensions of biodiversity are affected by edge effect, which is one of the primary outcomes of deforestation and habitat fragmentation. We want to understand the relationship between the animals that are left in these forest remnants and how stressed out they are. And to further that, we want to see how stressed out populations or stressed out individuals are more susceptible to various types of infections, diseases, and different types of parasites. And as part of that, we also want to understand how improving the connectivity of the forest remnants that are left, how that could help to ameliorate the overall health of populations in these forest remnants. This is what we typically see now in Paraguay. One of the things to consider is that a lot of this area is highly deforested, and you can see forests that are surrounded by primarily soy farms and plantations. And as part of that, this is what we find now in Paraguay. We're primarily working with small mammals because they're unique and awesome models to look at deforestation, ecological type analyses and models. This has included lots of prior field research, which has allowed for a lot of collections of specimens. Right here, I'd like to highlight just one person right there on the top. This is a young Lourdes Valdez, who is now a postdoc here with us at the Field Museum. This is a shout out. I really like this picture, this figure, which is from a publication we had last year that almost perfectly outlines and visualizes what we want to do. Big picture, what we want to do is we want to start to see the relationship between deforestation and how that affects the communities, especially along edges of forest remnants, and how that eventually will help, will affect the stress and disease that's found in various forest remnants that are left there. As part of this, we're going to require lots of museum vouchers, as one of the big problems that we've always had is identifying many of these specimens. As you see here, many of these specimens require that we collect vouchers because it's only through cranial dental characteristics that we've been able to identify these things. Many of these vouchers are already in the Field Museum, but we plan on expanding that considerably. We've been able to incorporate lots of students who've been helping with many aspects of this research already in the past. And as we have been pointing out, cranial dental characters are good ways to identify these things, but more and more we still need DNA. So big picture, this is why I like this figure, because this points out the importance of voucher specimens and all the various components of a voucher specimen that help us to then look at the infections of bacterial, ectoparasites, protozoans, and hopefully various, at one point, maybe even microbiomes. This is important because this helps us to understand the overall health. One of the things that we found is that towards forest edges, as we see right here, we tend to see an increase in overall biodiversity. That's the number of species, but also of those individual species that are there, the number of individuals. This is extremely important because this helps us to understand the community level aspects of landscape fragmentation. And here, when we start looking at landscape, one of the first things that we have to do is understand what's happening at the landscape level. This is a paper from last year where we're able to look at land cover change from 2000 to 2019. And as part of this, one of the things that we saw here in yellow, you can see all the areas of Paraguay, Eastern Paraguay, that have been lost just in the last 20 years. As part of this, then we're able to identify areas that are good for connectivity. And we're able to come up with models that allow us to understand connectivity for specimens that are very small, like small terrestrial mice, to medium-sized carnivores, bats, and then more charismatic megafauna like jaguars and stuff. And this allows us to come up with models of potential areas that allow for connectivity. Now, as part of this, we want to get to a more micro level, and this is where the zoonotic disease aspect comes in. This is from a paper that we had last year, where we were able to find evidence that populations of small mammals that are found in smaller forest remnants tend to be more stressed out. Our next part of this, and this is where we hope that we can tie everything together now, is we want to continue sampling. We want to capture various size forest remnants. We want to start measuring various glucocorticoid concentration in small mammals. And hopefully we can couple that with various viral, bacterial, and ectoparasite infections. That's the big picture of what we're going to do. We're just at the beginning of this. We want to thank lots and lots and lots and lots of people who have helped out in the past here at the Field Museum as well for the past like 8 years or so. And more than anything, I want to thank these little dudes. I want to thank my collaborators, Sara Boyle, who's at Rhodes College, Nancy McIntyre, Texas Tech, Pastor Perez, who's in Paraguay, who's going to be the main contributor in Paraguay, Lourdes Valdez, and Felix, who are here also at the Field Museum. Also, I want to thank all the colleagues here at the Field Museum and Mammals who have always been exceptionally helpful. There's too many to point out, but you could read them here. Thank you very much. And we're out.
Awesome. Thank you so much, Noe. And thank you to all of our presenters. It's thrilling to be here and to hear all of the great work that's happening. It's exciting to hear them and to know that they're happening in parallel. I'm Amy Rosenthal. I'm the director of the Keller Science Action Center at the Field Museum and a member of the steering committee of Chicago Wilderness. And so with bringing together these communities of scientists and conservation professionals and that overlap, for the Chicago region, we thought it would be worth having a panel discussion, questions, comments, ideas from all of you. Because after all, as the Walder Foundation has made evident about these awards, These awards help us to advance the frontiers of knowledge about biodiversity here and around the world, and also what we can do about it here in the Chicago region by working together. So bear with me as I add a spotlight to our other presenters. And as I do so, I will also ask a first question, and then I will look into the chat to see what other questions have arisen. So my first question is kind of the visceral. What is a day in the field like for all of you in the work that you're doing? What does it feel like, smell like, look like, and what do you love about it? And maybe I'll just ask you to go in the order that you talked. Give us a little bit of a snippet, feeling of the day-to-day. Adam.
Sure, thanks. It's a wide variation, so it depends on if I'm in the field or in the museum, right? So smells are always a rich part of it both ways. So earlier we were moving brains out of the skulls of animals that so that is not such a good smell. But the field work is, an amazing part. And I think the coolest thing for me has been realizing how excited I can get about the local biodiversity, right? I tend to go to far-flung places looking for animals, which I still love to do, but it's been really neat to, even short-tailed shrews. And I'm excited about seeing voles. As a Texan, where I grew up in the desert Southwest, we don't have a lot of voles, so they might be boring to others, but that's probably my what I'm looking most forward to, which people from this area tend to laugh because voles are really common, but to each their own, right? So yeah, I'd say it's rich in sounds and smells and touch and not so much taste, but yeah, I just like being out there and embracing it and experiencing it all.
I guess we'll go next if we're going in order. So I'll start by saying for me, and this echoes a little bit of what Adam said, I love being able to just get in my car and go to a field site and do some work 30 to 60 minutes from my house and not have it be a big production where I have to go really far away. It reminds me a lot of what got me into herpetology as a kid, which was just going out the door of where I lived in northeastern Pennsylvania and just going down the street and catching snakes or frogs or whatever I was doing. So I absolutely love that aspect of it. And also I'm getting to see and getting to know all the reptiles and amphibians of Illinois on a, very personal basis. I've never seen fox snakes until I moved here, and now I've seen a bunch of fox snakes, which is very exciting to me. So for me, that's one of the things that makes me really excited about this project.
Yeah, I guess I'll just talk a little about the day-to-day. Fieldwork is fieldwork. It's what I love about it is you're muddy. Sometimes we're wading through a pond. Sometimes you're in the middle of a grassland. Also, as Adam mentioned, there are a rich array of smells. We have a lot of garter snakes at our site. And if you've ever picked up a garter snake before, it sort of musts all over you and that smell sticks with you for a long time. And also what I've really enjoyed for this project, because our interns, once they're trained up, they're really the ones leading it, is when I get to a field site now and I'm out with interns, I'm like, all right, guys, you lead. And I'm just kind of like following and I'm helping where needed. But it's like, they're taking me on this fieldwork journey. So it's a little bit different, and it's fun to watch them take that lead.
I guess I'll go next. I think I'm very fortunate to be able to use this to actually go out to Paraguay, which is a place I've been many times before. I am extremely pumped. I think hopefully soon this semester we're gonna have our first trip out to Paraguay. I've been there since 2017. So it'll be pretty awesome to go back and be in the middle of the forest. For anybody who's ever been in tropical rainforests, tropical rainforests are remarkable. There's nothing like that. When you're out there, it's like you're alive when you're walking through a forest, right? Just sitting there and just hearing, every once in a while you get a chance to just stand there and just be able to hear all the living things that are around you. That's pretty remarkable to me. That's what got me into mammalogy to begin with at one point in my life. And, I've never regretted that decision. So yeah, it's an amazing place.
So there was one question early in the chat that I saw from Abigail Derby Lewis asking if any of the sites of folks working in the Chicago region are Illinois nature preserves. And then I also see one from Nigel Pittman. I wonder, Nigel, if you wanted to unmute and ask your question.
I'll just chime in and say yes, for us, some of the sites that we're at are nature preserves. You know this because you can't bring dogs to them so reserves versus preserves. So there's nature reserves and some of our sites are there. I don't know if there was a follow-up on that, but the answer for us is yes.
Yeah, so that's wonderful. I actually chair the Illinois Nature Preserve Commission, and so I have a deep interest in this. And part of what makes a preserve so special is presumably there is very, very high quality, often remnant habitat in these areas. And so my follow-up question is, if in fact you have a mix of nature preserve sites and non-nature forest preserve sites, I would expect you might see some differences in her community, potentially even population density, but I would be really curious. Did you see some differences, or what were sort of the similarities and differences, I guess is the question.
So as of right now, I don't think we can speak with any super certainty because we're still in the process. This field season is still running. So we haven't collated all of the data points and put them through any sort of statistical analyses to see if there's more diversity or abundance at certain sites versus others. I can say off the top of my head, the places that are more managed with respect to doing burning, whether that is a preserve, a nature preserve or not, are sites that have, I'm going to say, for lack of a better term, better herps. They're things that are more associated, at least with prairies.
Thank you. And I really look forward to hearing how the rest of the season goes. And we'll talk to you offline. I think the commission would really like to hear perhaps next year about these results. Thanks so much.
Oh, our pleasure.
Nigel, did you want to unmute?
Sure. I have a similar question, but it's about time rather than space. I was just wondering, when you reread these old surveys, you got kind of an idea for what a site would be like. What were the biggest surprises that you got visiting the sites now, things that you weren't expecting based on the old surveys?
I'm a little surprised that we haven't come across any of the invasive stuff like red-eared. I mean, red-eared sliders are sort of native to this part of Illinois, but at the same time, in many places, they take over sites and they're invasive worldwide. And at least for our turtle surveys, I'm a little surprised we haven't come across any. We've been getting a lot of painted turtles, which to me is an indicator of, I don't know whether I'd say higher quality habitat, but more in balance than when you're overrun with these red-eared sliders. The other thing I'll say that we've seen is that in some sites where only one of a species was originally recorded, like smooth green snakes, for example, there might have been one only recorded over several years, but at those sites, which are also some of the more managed places I just mentioned to Abigail, we're seeing a lot more of those animals than we're seeing maybe 10 years ago in those more managed, prescribed burned areas. So that's actually really nice. On the flip side, there's things that we haven't seen that have been recorded, but they also might not have been accurate recordings to start with.
So stay tuned for more, because more about temporal changes and spatial habitat integrity changes are going to come out over the next couple of years from Sara and Michelle. I think that Leslie de Souza actually also had a question in the chat. I wonder if you'd unmute and ask your question to Noé.
Hi, everyone. Congratulations to you guys. It's so great to hear the work that you're doing. So thanks for sharing it. I learned a lot and excited to hear kind of where these projects, the results of some of these projects. Noé, I was so interested and curious about some of the new results you found, and just that these little guys are all stressed out in these smaller fragments. But was curious if there was any prospect for creating some kind of conservation corridor. The models were so clear that you had some options for what that could look like to improve the health of the forest, but also the species that live there. And I was just curious if there was conversation with government or partners or organizations in Paraguay for that.
Thank you for that question. I appreciate it. That's pretty awesome. So that's actually really interesting that you mentioned that. I could show you the map later on, but one of the things that popped out of that analysis was that many of the forest remnants that would be very valuable for connecting the Atlantic forest in eastern Paraguay are outside of this proposed biological corridor system that's happening already in paper. I mean that in paper because there's all this funding that's been provided by the World Bank, and they're doing all this reforestation. And interestingly enough, none of the areas that we found to be important for connectivity are part of that process right now. And so it's interesting to think about when all this money and effort is being thrown at things like reforestation, how come we're not doing more analyses like this now? There are some potential things that we've been talking about, because where we're going to be doing our sampling is right smack in the middle between big conservation areas that are found there right now. I think if we can make some kind of an argument beyond just conservation per se, and maybe we start putting a zoonotic disease. I think with COVID, it's very obvious that we can now relate this to the general public and be like, see, this is what happens when we have more and more deforestation and we don't take care of our environments, right? I mean, I can't put it more straightforward than that. And so hopefully there are many potential zoonotic diseases in the area, even things like hantaviruses that are known for that region, among other various rodent-borne diseases. And hopefully as part of this, we could say, look, these things are more stressed out. They're living near people's farms and houses. If we don't reforest this area, we're just setting up for the next pandemic. Hopefully, I think there's one way that we can start looking at conservation and other things from a different angle, and be able to work with the vertebrates that we like to begin with. So, yeah.
Yeah, I actually wanted to add on to that, or build from that question, because Noe and Adam, you are both taking a One Health approach. Could you tell us a little bit more about what that is and why you think it's important? You're doing it in really different places in the world, but I think hoping to answer some of the same questions.
Would you like to go first, Hannah?
Sure. Are we doing the order? Yeah, sure. That keeps it thing. Yeah, definitely. So the One Health is not a new concept, but the idea that human health is tied to environmental health, which is tied to animal health, right? So human, environment, and animals, we don't exist in a vacuum. Obviously, COVID has really brought that One Health approach to the scene, but we tend to do triage, right? One Health after the fact. So I think one of the benefits of this is trying to set that up beforehand, and museums are in particular well equipped to handle that because we're biorepositories for samples and specimens, right? And so you can collect them pre-pandemic, so you're able to go back and look at what happens and look at these changes. And I think one of the I had a really important point I wanted to make about the One Health approach, and I totally lost it. I think one of the things I like about it most is it's integrated by definition, right? So it requires that, oh, I remember now. And so we do a lot of One Health work in Kenya and East Africa, for example. And one of the questions we get is like, well, how do you implement this back home? And we're kind of like, we don't, right? And so I think this has been a really cool way to say, like, look, Lyme disease may not be Marburg or Ebola, right? But it is a huge problem, and it's a huge concern for people that are using these systems. Raccoon roundworm is a lethal pathogen that's transmitted through raccoon feces, right? Which is really common in places. So I think it's a really cool attempt to try to practice what we preach in other places in our own backyard and see if it helps, improve our humans' well-being and protect humans' health and animal environmental health in this system.
You're muted.
There you go. Then now? You hear me now? Okay, sorry. So I think one of the interesting parts about this, right, is that we get exposed to a lot more of these potential pathogens more than we think. So one very specific anecdote that I would like to point out, a few years ago, we ended up starting a project in West Africa, in Ivory Coast. And a friend of mine asked me if I would collect some blood samples from the specimens for screening of hantavirus. As we were doing that, when you're doing field work, I accidentally pricked myself with some, wire or something, and I started bleeding. And it occurred to me that I should probably put my blood just to screen it, just because of all the field work I've been doing in the past. And so I ended up putting some of my blood in one of those little slides for him to process. I sent him back to him. He gave me the results of the specimens that had hantavirus, and he said there's one specimen that's got more, it has more presence or evidence for hantavirus than any of the other samples, and it has your name on it. What is that? And I was like, that was my sample. And so it's interesting to show that we've, especially as field biologists, we've probably been inoculated by all kinds of viruses and pathogens that we don't even think about. And that kind of shows that we are exposed to a lot more pathogens, especially in tropical regions, that we realize. And usually, many times, it doesn't, well, you either die or you end up just getting past it, as we've seen with COVID. But one of the things to keep in mind is that this is everywhere. This is all around us, you know, and it's interesting that we quantify the amount of stuff that's out there, especially in the tropics, where we don't even know the mammal species or the vertebrate species, let alone the viruses they carry, let alone the other bacterial infections, the ectoparasites. And that, to me, is a really cool part about this, is that, like, as Adam was saying, this is a very multidisciplinary thing, because we were going to have to collaborate with people who do ectoparasite work, with people that are going to help us with some of this viral work, and just part of it now. You know, you can't do it all, you know, or else it's just, that's part of how it goes. Oh, the other thing to keep in mind, I just want to throw this little pitch in there. Like, this is an idea that I've had literally for almost 15 years, which is originally what I wanted to do. And thanks to these funds, which are a good chunk of change, we're going to actually be able to actually get it done now. Well, hopefully try to get it done. So that's a cool part about this type of funding, I think.
And there was a question earlier. I know, thank you, Sarah and Michelle, for responding to Laura Bargusen in the chat. Laura, did you want to unmute and say anything about Bobian Woods and kind of listening there, or ask a follow-up to Sarah and Michelle?
Thanks, Sarah and Michelle, for the feedback about the wood frogs. We're getting ready to do some prairie restoration there. And there's just like a little path that some interns made through a wooded area by the little Calumet River. And so far, just being there, you know, doing mapping and pulling of invasives, we saw a smooth green snake, and we saw this frog, which admittedly was going by really fast, but to me, it looked like a wood frog. And so we've been thinking about how, you know, what would be sort of the easiest and most efficient way to do some wildlife monitoring down there to get a sense of what's there. And then hopefully going into the future, you know, after the prairie plants are installed, you know, to see, you know, what happens after that. So that was the origin of my question.
Yeah, I guess I'll follow up on that. Is audio recordings are a great way, because it's really hard for people to like always be there, right? They're not going to be there 24 hours a day, like standing in the area you're interested in. So audio recorders are a really nice way to try and ID if species that are hard to see are there, but also gives you something that you can store and compare to over time, right? Because you can always have those audio recordings. So it's a nice way to see change over time, and in a way that might not be obvious just with visual surveys. And then I'll just mention that one thing that's come up in audio research that's really allowed more people to engage in these types of recordings and bioacoustics are reduced price of the recorders, right? So they used to be like several $1,000s, and then, you know, that really limits to who could like have access to these sort of things. So recorders like Audio Moths, which are more like $60, are a lot more affordable, and so it's really opened up the door to this type of research, and also open access software where you can actually sort of like go through the recordings. I think has really like launched these sort of research questions. So feel free to reach out if you have any more questions.
Yeah, thank you. Yeah, I was talking to Becky Collins at the Forest Preserves of Cook County, and she was mentioning the audio moths as well. So that's probably what we'll look into. And I probably will contact you about the open source software as we progress. Thank you.
Awesome. Well, that's a great connection. We have one last question in the chat that we have time for before Jack Westwood helps us to conclude this webinar. And it's from Jacob Campbell. Do you want to ask your question out loud, Jacob?
Yeah, sure. Thanks, everyone. Really inspiring work here. I was just, my question is related to students in your projects. It seems like more than one, or if not all, in some way, there are young people up to postdocs involved. And I was just kind of curious if you could, anything surprising or of interest that they have brought to your project so far, or like looking over the horizon, or roles for them that you're excited about.
Sure. Yeah, I think not related to the project, but today I learned one of the Zoe doesn't know what the movie Gremlins or Gremlins 2 is. So that was yeah, that was a shocker to me. So no, I think one of the coolest things about this and these integrative projects with the students is we've got so many different kinds of partners that are doing different kinds of work. So there's, one of the things we always try to do is tailor, we have objectives that we need to meet for the project, but also like, what are they interested in, right? So if someone's more interested in the museum component, they could do that, or the field component, or they want to do administration or stewards or conservation or sociological aspects, like that's a really great thing. And so I've been amazed at all the students that we have, how they want to get experience with everything, right? So they're like, oh, I do want to, like I came in to trap the rodents, but I really want to do the museum databasing, right? Or these types of things. So they're really kind of hungry for any kind of exposure and experience, which is a really fun group to work with, right? Because you're like, oh, great. Like, yeah, so.
I'll just echo what Adam said again, which is the students we've been working with, the interns, the field interns, they in particular are so amazing, have done such a fabulous job. I don't want to say I'm surprised by how great they are, but to some degree, it's surprising how quickly they were able to go from not having done stuff like this before to being the leaders of those teams. I guess what I'm really not surprised about again, but two of them have now started volunteering in their off time in herpetology here at the museum so that they can do other stuff like databasing and entering data and working with the collection. They wanted to get both sides of things. I'd probably thank Josh for exposing them to that, because he gave them a tour and it made them get super excited, not just about the field work, but also about some of the museum collection side of things as well, which is awesome.
Yeah, and I'll just quickly say one of the surprising things to me is I went to one of the field sites one day that I had never been to, but the interns had been to many times. And it's one where it has very accessible trails. So there's a lot of community members there, and a lot of people in the area, in the neighborhood were walking and stopped me, and they were asking me what I was doing, and I explained, and they're like, oh, we usually see these other two girls here. And when we talk to them, they show us the frogs and the snakes. And that happened to me 3 times when I was at the site with like 3 different people just like walking the trails. And so the interns, it was interesting to me that the people like called that out, that the interns are engaging with the community as well.
Yeah, and I mean, right now we've not been able to get this project started yet, but historically, with all the students that I've had, just generally speaking, it's interesting to see how getting exposure to these types of fields, I think we forget, right? We think that everybody loves being outdoors and everybody likes this type of research. Many students have never been exposed to this types of research, and being exposed to these types of experiences is a big pathway for many people to experience things they never even thought they wanted to do. This is especially important with underrepresented students, people who we can look around are not represented here, especially at the higher levels of STEM and academia. And so this is a perfect pathway to diversify our fields. And once you get people out there, especially holding like a snake or a frog or a small mammal, this is a game changer for lots of people, and it starts, opening avenues that they never even thought about. So, and even in people who may not become biologists down the road, who become, business majors or whatever, it's important for them to understand that this applies to all of us, and we can't just, we need to expose everybody to these things, because at the end of the day, it's where we live.
So It has been a real pleasure just to share a few moments of the stage with all of you. Adam, Sarah, Michelle, Noe, you guys are doing incredible work. And this community, I know, is one that really cares about the kind of science that you're doing, the connections that you've just described that are being made with communities, and the education that's possible, and the potential to diversify the environmental and sustainability fields here in the Chicago region. So thank you so much for being with us, for sharing your work. Please come back, please share what your updates are. And I'm going to turn it over to Jack to see us out. Thank you so much. You've been an amazing audience. Wonderful questions. Thank you for joining us today.
Thanks, Amy, and thanks everyone for attending today. Thank you to Chicago Wilderness and the Field Museum for hosting this. It's been a really great session, and I think Chicago is so fortunate to have these, this excellent community of biodiversity researchers here that are committed to, turning their research into on-the-ground results. We've heard so much today about these great partnerships, how Chicago expertise is helping to tackle the biodiversity crisis overseas, and also how experiences from elsewhere are being brought to bear for conservation outcomes in Chicago. So it's been a really great session. Just a reminder that the Biota Awards are now open until next month, October the 3rd. So I'm really excited to see the proposals that come in this year, and I'm sure we'll have another great group of awardees to talk about next year. So biotaawards.org is where you can find out more, and please be in touch with any questions. So thank you to our speakers, and thank you also to the two awardees not featured today as well, Becky and Megan. Thanks everyone.
Thank you. Bye everybody.